Hello, I'm Dr. Gary Zammit, President and CEO at Clinilabs, a full-service CRO that specializes in the development of central nervous system therapeutics. And with me today is my friend and colleague, Dr. Michael Detke, MD, PhD, who is president of Detke Biopharma Consulting and clinical professor of psychiatry at Indiana University School of Medicine. Welcome.
Thanks, Gary. Delighted to be here. Thanks for inviting me.
Now, many people may not know you graduated with BA and MS degrees in psychology from Yale, and then later received your MD and PhD degrees from the University of Pennsylvania. From there, you went on to Harvard Medical School, McLean, and you've since published over 70 manuscripts in peer-reviewed publications, and you've been involved in the industry for over 26 years involved in CNS clinical trials in many major illnesses. So quite a background that you've had.
Thank you. Like Clinilabs, which focuses only on central nervous system disorders, I'm a psychiatrist by training and have focused my entire career on CNS clinical trials and development of therapeutics. So it's been a nice match.
What led you into this career?
I always wanted to figure out what makes people tick, and clinically, I found psychiatry to be much more interesting from a doctor-patient relationship than some of the other. A series of migraines might be the same for almost every patient, but as you know, Gary, as a fellow practitioner and researcher in the field, every patient you meet with schizophrenia has a different story, how it's impacted their lives, how it presents. So I always found psychiatry to be endlessly fascinating and an area of great unmet need, as you know. So an area where one can make an impact to help the world a little.
Do you think we have a way to go?
I think we have a long way to go. As you know, some of the highest unmet needs in the world in medicine are in major depressive disorder, schizophrenia, suicidality, and Alzheimer's, Parkinson's, going further afield to neurology. But we've also made some great strides recently. So in the course of my career, 26 years now in the field, we've come out with some great new treatments, non-stimulant drugs for ADHD. Just a few months ago, an approval of an absolutely new therapeutic mechanism for schizophrenia, the M4 PAM, M1 M4 agonists and PAMs. So there's a long way to go, but a lot of progress and hope for the future.
One of your areas of expertise is in placebo response. Let's spend some time talking about that. That's really the focus of our conversation today. What are placebos? Let's start there.
Yeah, it's a great place to start. Placebo is from the Latin. I will please you or mollify you or calm you. And it's, a placebo is an important way in that we test drugs and other treatments. You have one group get the treatment with the actual drug in it, and the other group gets a placebo pill, a so-called sugar pill, which does not have the active drug in it. Or in device work, you use a device that, say, stimulates the brain, and then with the control group, you have a placebo equivalent, or a sham, as it's called in devices, that doesn't deliver the key brain stimulation or whatever else it might be delivering. And there's another differentiation I think it's useful to make at the beginning of any discussion about placebos is what's your definition, my definition of placebo, because one way to split it up is to talk about what you think underlies the neural networks underlying the placebo response in our brain, how placebo might be happening, how it might be working, where in the brain it might be working, etc. And NIMH just did a nice symposium this summer where they spent a whole day on that topic, and then half a day on that topic, and then another half a day on just the practical aspects of placebo response in our field, which is clinical trials. And those practical aspects are what I've really spent a lot of my career focusing on, how to diminish the placebo response and then to really improve drug placebo separation or signal detection. So I think as mature drug developers, we should talk a little bit more about signal detection, because what you're really trying to find out is if the drug works and give it its best opportunity to show you in the trial that it works. And that means separating from placebo.
What constitutes a placebo response?
Again, so you can answer this a little bit more about mind over matter, or subjective expectations on the part of the patient and or the clinician. But to us as drug developers, the simple definition is, how well did the placebo group do? So if you have a schizophrenia trial where the drug makes the patients 12 points better and the placebo makes the patient 7 points better, that's the placebo response. To me, the important question, that 7-point placebo response, is what really matters. And it's, so if that's a big enough difference, that delta of 5 points in my example there, then you'll see a significant outcome in the clinical trial. And if not, you won't, and will have wasted a lot of time and money, maybe testing something wrong, or maybe testing a drug that isn't very efficacious. So the placebo response can be a real challenge in drug development, as you know.
And a placebo control is really standard in clinical trials today. That's the gold standard, really. How many trials succeed or fail because they've got a placebo included in the design?
So just go back for a moment to Science 101. You try to control everything. You try to have two groups of people with the same amount of males and females and the same ages and things like that. And you do that by randomizing, which does a pretty good job of ensuring that you have balanced groups. And then you control everything except the one thing you want to test, which is the drug versus not, or something like that. And the placebo response varies in a number of ways: duration of the trial, the therapeutic area or illness you're testing. But one well-known answer to your question is that of the currently available approved drugs for major depressive disorder, the antidepressants, of all the trials that all the makers of those drugs had submitted to the FDA, about half of those clinical trials failed to show a statistically significant difference between the drug and the placebo. Does that mean antidepressants don't work? No, it means they clearly do work, because they work in many trials, and being able to show a statistically significant difference between drug and placebo allows you to make a strong scientific conclusion that there's a difference here. And when you don't see a difference, you fail to reject the null hypothesis. So you have failed to prove that they're different, but you haven't proven that they are not. You just failed to prove it. So those positive trials give us strong confidence, and that's held up in many other meta-analyses and real-world trials of antidepressants and antipsychotics and so on and so forth. So the challenge is that the effect size may be modest. The effectiveness of the drug, I think, would, as lay people would say, can be modest. And then if you have a little bit of a placebo effect too, it's hard sometimes to show whether the drug is really working or not.
And that rate of failure to separate from placebo of about 50% that you've cited in depression, does that also apply to other therapeutic areas?
You see some placebo response in almost every therapeutic area. So for example, it is staying within the therapeutic area of psychiatry, but looking at schizophrenia, the failure rate there is closer to about 25% than 50% in recent years. So the placebo response takes different forms in different diseases. Recent literature and recent trials that have read out in drugs for Alzheimer's disease, going now to neurology, have shown a pretty significant failure rate too. But the final report on that hasn't been written, because there's still new mechanisms that are being tested, and we don't know if they work, how well they work, etc. But as an example, I like to take from kind of extremely disparate therapeutic area. There was, I think, in a Houston VA hospital, they tested surgery on knees for knee pain. And there's a standard surgery that surgeons do and kind of clean out the knee joint. And they did a sham controlled experiment where some people got an incision made and they cleaned out the knee joint, and other patients didn't, and they weren't told. And it didn't work. It didn't separate from placebo. The placebo effect was, or the sham effect group did as well as the controlled group. So the placebo response, and I have slides from talks I've given that show placebo response in many different therapeutic areas.
In some of my reading, I noticed that in pain trials, that when one placebo tablet is administered, there is a placebo response. But if two or more placebo tablets are administered, there's even a greater placebo response. Is that an unusual phenomenon? Do we see that in clinical trials today?
I like to use the word salience. So it sometimes helps to, if there's some sort of, imagine a bond forming between the drug and your knowledge of its efficacy, how it's going to help you reduce your pain or whatever. It's sometimes helpful if the pill or the signal, if you will, is highly salient. And by that, we mean something that is clearly observable, kind of, brighter colors. There's some literature like what you described that in different areas that like a red pill might work better than a green pill, all other things being equal, like not actually having the drug in it or being placebos. And one other kind of interesting area that's sort of closely related to this is that, drugs that have side effects can do what we call functional unblinding. So drugs like the antipsychotics on the market now that mostly cause increased hunger and weight gain. Well, if you're a patient in a double-blind placebo-controlled trial of, say, Zyprexa, then you're probably, if you're on Zyprexa, you're probably going to get hungry. And if you get hungry, you're probably going to think you're on Zyprexa. Same thing with drugs like the psychostimulants for ADHD. They can cause sweating and increased heart rate and a stimulanty feel and insomnia. So again, these salient cues are very important in how we test the drugs for optimal effectiveness and how we design our trials and implement our trials to be as effective as possible at trying to find that signal detection.
Yeah. So the cues can promote a placebo response, but how do we deal with that in clinical trials where we use double blinding, right? So the investigator and the patient neither knows whether or not the patient is getting active treatment or placebo. But how do we control for that placebo response? How do we control for those cues?
You can't really get rid of it. But that's kind of a good thing. Because so again, back, Science 101, we try to control the cues on both the drug and the placebo and do that equally, and just control the drug. But here's the thing. This is an important point that it took me a long time to consider. So let's say you've got your drug effect, which is big, and your placebo effect, which is less big. Okay. And now let's say you do something that in theory increases the placebo response or the response to both. In theory, they should both go up like this and not make it any harder to detect, right? But in practice, what my two hands here are a mean of 12 point and a mean of a 12-point improvement on drug and a 7-point improvement on placebo. But in reality, what you have is a lot of noise. Those are the averages of the two groups. But you've seen spaghetti plots, and most of you out there listening to this have seen spaghetti plots, and there's a lot more noise. So if you don't do everything you can to control that, you're just going to get more noise. And again, signal detection is always signal to noise detection. It's really just a question of whether this difference is a lot bigger than the variance, and then you've got a drug that works. And if the difference isn't bigger than the variance, then you don't have a treatment that works. So anything we can do to decrease the variance is important. So we do that by trying to enroll patients above a certain severity of illness. And in general, in controlled randomized trials, you want homogeneity. You want everyone to have the proper, correct diagnosis, the same diagnosis, roughly the same severity. You start out by testing only in adults because kids would add more variation to it. And so on and so forth. All those things in all those long lists of inclusions and inclusions are the ways we try to minimize variance, minimize error variance, as the statisticians call it.
You mentioned that we want to look for subjects to enter into these trials that are kind of homogeneous, right? And they you want to have a minimum severity of illness. But um, if all people are getting treatment, whether it's active treatment or placebo, and then the same rater is rating, and they know that the person is getting something, so they tend to favor showing an improvement because they know that the patient is getting something.
You're talking about expectation bias. And there are nice studies that show that the patient is expecting I have in a typical trial at least a 50-50 chance of getting some active treatment. And the clinician expects that there's a chance that they could get better. And in fact, there's some data to show that there can be interaction between the clinician and patient that also pushes in this direction. There's a little bit of research on databases in both depression and schizophrenia that support this concept further, in as much as, in some trials, you might be testing just drug and placebo, and you might have 50-50 randomization. So you have a 50% chance of getting the active drug. But in other studies, we might test maybe three doses and a placebo, and you might do equal randomization there. And now if I'm a patient in that trial, I've got a 75% chance of getting drug. And there's some evidence that expectation does increase the placebo response a little bit.
What about centralized rating? I know that you have had quite a bit of experience in that area, and you've been involved in kind of developing processes for centralized rating. How does that help with the placebo issue?
To talk about centralized rating, and we're talking about rating of clinician-rated subjective scales like depression rating scales and schizophrenia, symptom severity scales, and so forth. And if you're going into some new area, it's nice if there are some analogies you can use. And there very much are in our field, in as much as, we've been using centralized labs and centralized image reading and things like that for years and years. And they're actually highly recommended, if not almost required, by major regulatory agencies. And these are so everyone gets their test tubes full of blood sent to one central laboratory, and that central laboratory has one machine that they run the assays on, so they get very consistent assays, as opposed to sending it to 40 other labs and having different reference ranges and all that mess. And centralized rating in clinical trials is done similarly, except as Gary and I are doing right here, the clinician and the patient meet via video conference or audio conference, depending on various parameters. And so you can have a small number of raters, all of whom are trained and consistently trained against each other to administer the scale the way it was designed to be administered, and as accurately and consistently. And part of it's just consistency. So what we found, I've been involved in that for years, and what we found is that we think it affects two important things. One, and I've already kind of implied this, is variance, right? If you can take a site with 40 trials, with 2 or 3 raters at each site, maybe 100 raters, and do it with 10 or 20 raters that are centralized, and you can just do video conference to the patient site. That decreases the variance, which is half the battle. It's signal to noise, and you just decrease the noise. The other thing we found, though, is that there's a lot of bias, and we were talking about that a minute ago when you talked about expectation bias. And there's a lot of bias on the part of the patient. The patient expects they'll get better. The clinician expects they'll get better. And in fact, we kind of shoot ourselves in the foot in a sense that we require that patients have a certain severity of disease, and you have to have a certain severity to be able to see improvement. But we recruit patients when they're at their worst. And there's something called regression to the mean that's a very powerful phenomenon. It basically means if you're an outlier that's high on some scale or an outlier that's low on some scale, over time you will gradually regress to the mean. There's a classic example of this, which is just You find a new favorite restaurant. You went there once and it was fantastic or terrible. Almost always, if you go back there, it'll regress to the mean. It won't be. It was fantastic that day, maybe because you were really hungry. Maybe they had their best chef in that day. Maybe it was just, the person you were with was particularly entertaining or something. And that hurts us a lot. Bringing it back to a clinical trial, you have all these people that are way up here, and then they come way down throughout the trial. So instead of having an effect of 12 with your drug and 7 with your placebo, maybe you have 22 with your drug and 17 with your placebo, but again, with a lot more variance. Those are a couple of the reasons why central raters can reduce bias and can reduce variance.
Yeah. So, no, I think this is an important point, because with regression to the mean, it really doesn't matter if you're on placebo or not, as long as there are kind of multiple assessment periods. The later periods are probably going to regress to the average, regress to the mean, whether somebody's on a placebo or not. But if they're on a placebo, they could be regressing to the mean and have some other placebo response on top of that that makes the placebo response sometimes look pretty good.
No, that's exactly right. It's if you enroll patients when they're at their worst, high symptoms, then whether they get a placebo or not, they're only going to get better. There are different placebo response magnitudes across different kinds of trials, and this one, this one makes the point that if you're treating patients that are sicker, you might get an even bigger placebo response. Now you can get patients who are treatment resistant or chronic, and then you may get a smaller placebo response. Regression to the mean tells you that all other things being equal, if your trial is longer, you're going to have a bigger response because they're just going to keep regressing to the mean. So you might have you might have a bigger placebo response in the beginning, but a shorter trial might give you a slightly shorter placebo response. It might give you a slightly smaller drug response too. And there's always a double-edged sword to everything you do in clinical trial design. You lower the placebo response, you might also lower the drug response a little bit.
Are there biomarkers that might help to identify those who are more likely to respond to placebo than not?
Yeah, absolutely. Biomarkers, wherever we have a good, valid biomarker in the CNS space, it's our friend. And for lots of reasons. I mean, the biomarker might tell you, you see changes on an EEG, and at least it's getting into the brain. So that's good. You see changes in a system downstream, and you have even better evidence of that you've got that you're hitting your target. You've got target engagement. Probably the best for CNS drugs would be if you've got a PET ligand. And here the other thing is those 50% of all those depression trials that failed. Well, you have to put it in the context that this was in development and they didn't know. We know what the right dose of Zoloft is today, but the Pfizer scientists that were working on it at the time back in the 80s didn't know the right dose. So for example, PET ligands can help you narrow down and get the right dose. So remember, if you got the dose right, you can fail 50% of the time. If you have the dose wrong at half of the right dose or a third, that can make getting those trials to seeing that signal detection, that separation, all that much harder in the trials. So yeah, biomarkers, whenever we can employ them, are our friends. They're often very idiosyncratic to the target, like a PET ligand. But there are definitely some things we can use they're not specific, like EEG changes and a few other things.
Can they distinguish, though, between who might respond to placebo and who might not?
Boy, that's the million-dollar question. If you had a biomarker that can demonstrate a difference between placebo responders and not, and mostly I think that has not been done yet. There are some interesting companies developing EEG-based biomarkers now and using things like machine learning to identify differences, and have some encouraging early data that we might be able to better identify placebo responders.
Yeah. I'd like to go back to the question of central rating. In central rating, are the raters blinded to the visit type that the patient is going through? So do the raters know that this person is in screening, or after only 1 week, or after 10 weeks? Is that part of what's done in central rating?
I think the way to think about this is, imagine if you're in a clinical trial office and you work there and you're a rater. And if you've never been to one, just imagine you're a patient in a doctor's office and you're one of 20 patients that go in and see the nurse or the doctor, and they ask you a bunch of questions. And when you come back the following week and the following week and the following week, it's probably with the same nurse or same doctor. And they ask you sort of the same questions and everything. And they know when you come in for your first visit, they know it's your screening visit or it's your first visit to see this doctor. And they know the next one is a baseline visit, and we know that you have to have a Hamilton Depression Rating Scale score of 22 or higher at the screening and baseline visit, etc. It is easier to blind remote raters to this. You just ask them, not to look up anything about this Eli Lilly trial, this BMS trial, whatever trial you're rating in. And they sit probably in a home office like I am in right now, and get videotapes sent to them all day long. And what all they know is at 9 o'clock I have to do a video interview of a patient for a schizophrenia study and rate them on the PAN scale, positive negative symptom scale. At 10 o'clock I have to rate a patient in a depression study on the Hamilton Depression Rating Scale, and at 11 o'clock you get the idea. And so their motivation is to just do that scale as well as they possibly can. They usually don't know if they don't know anything about the trial. They don't know if this is a patient in a year-long open label trial and they've been in for 9 months. They don't know if it's a screening visit for a 6-week trial or anything like that. So that helps to reduce any motivation or any bias to think, oh, this is a screening visit. They're probably going to be really sick because regression to the mean, right? But they usually don't know that it's a screening visit. There are lots of ways to do this, and there's some good data showing that way is pretty effective at improving signal detection. But there are other ways of doing it. One method that a lot of companies are using is having, if you're the clinical trial rater at the site, Gary, you do the rating at the site and videotape it. And then a third party might review a subset of those videos, maybe randomly chosen, maybe chosen to be relevant to the key questions like enrollment at screening and baseline, or if you see a rater not doing quite as well once or twice in a row, you might check that rater again on their next assessment. So there are several ways to do central rating or central review.
Ultimately, does central rating help to reduce the placebo response rate? Does it improve signal detection?
There have been about 7 head-to-head trials where that's been looked at with central ratings, and in 5 out of 7 trials, it seemed to improve signal detection or reduce placebo response, and in 2 of them, it was pretty equivocal. But again, as I said, when we discussed a different topic, there are more data coming in. There are more data. There aren't many data yet on controlled trials within trial of the reviewing the videotaped version. But there are a lot of data on that, so I'm looking forward to there being more publications by various companies.
Some sponsors collect data on the placebo response rates at specific investigator sites. And if they find that one investigator site is producing a very high placebo response rate, not separating active drug from placebo, they may be less inclined to randomize more patients at that site or to use that site again in future trials. Does that help to reduce the placebo response rate?
Picking the right sites is important for a number of reasons. You certainly want to be working with people that are doing their best to be, have high integrity, and job number one in every trial, every site, every patient, every day is patient safety. So ensure patient safety, good clinical judgment. That being said, the most important question you might ask of your sites is, can you separate drug from placebo? That feature is really hard to evaluate. And I think there's 2 reasons why it's hard to evaluate. One is because We do these trials of 300 patients because it takes 300 to have enough statistical power to show that drug worked better than placebo. So if at your clinical trial site you only had 10 patients, which is a good job in a 300 patient trial, right? 5 on drug and 5 on placebo, that one bad placebo response could make your site look bad there. But they're ridiculously small numbers. One good drug response could make your drug placebo separation look fabulous, and when it probably shouldn't. So the one problem is the small numbers, and the other problem is raters move from site to site and move on to different parts of their career. A lot of raters are, graduate students that might want to go on to, from their master's to a PhD or something like that. And the raters are really doing that. And so if the rater turns over, that part of your site turns over too.
If somebody was designing a clinical trial protocol today, say in some general area of psychiatry, what would be the top recommendations you would give to that person to help to control placebo response rate and maximize signal detection in the trial?
I think there are a number of tools to use. We've talked about central raters and central review. That's definitely one important tool, but it certainly should be fit for purpose. And I'll give you another tool, and I'll give you a couple of examples. Another thing we know is absolutely true about pills and testing whether they work is that they're only going to work if the patient actually takes them. So medication adherence is a problem in some trials, and duplicate subjects are another problem in other trials. And we've talked about this, Gary. There are sometimes patients that will sign up to be in a trial twice, maybe at 2 different sites. Maybe you've got a site on the north side of Cincinnati and a site on the south side of Cincinnati, big cities, whatever. Or they might be in multiple other trials at the same time. This could be just a lack of awareness on their part, although it really shouldn't if they read their informed consent. But sometimes people have motivations for doing these things. Sometimes these duplicate patients don't take their pills. There are again a bunch of different companies out there vending methods for make sure your medication adherence is stuck to. And you can draw blood and test the level of drug in the blood, obviously on the people that are randomized to drug at least. That's PK is one way to do that. There are other methodologies out there. I consulted for a company called AI Cure, which has an app that can do facial recognition and show that the patient, using machine learning, showed that a patient swallowed by looking at swallowing muscles and things. And of course, time and date stamps everything. So that's one, and they have data showing that correlates with actual blood levels of drug, which is why I like their methodology, because they got data on the gold standard. And so those things are important. And then, let's think about if you're applying it to a trial today. If you're applying it to a trial where you're testing psychostimulants, I would say that's an even more important time to use this to make sure that you don't have duplicate subjects, because those are drugs of abuse that a lot of patients might sign up to get those drugs, take them themselves, sell them on the street, whatever. And other things being equal, if you're testing Prozac in that same trial, that's not a drug of abuse. No one really wants to get high on Prozac because you really can't. So you have to think about the drug you're testing, what you're testing it for, et cetera. And then go to this toolkit of different methodologies like medication adherence, duplicate subject avoidance. There are several companies that do that. Centralized ratings or review, and there's more. But in today's age, when all of these things can be happening, and when signal detection is challenging in CNS, we want to consider all of this carefully. We've talked a lot about design too, and implementation is equally important. You've got to follow these sites, got to make sure that the ratings are being done well, consistently, and that's takes a lot of work throughout a trial.
Anything else you would like to add?
We covered this at the beginning, but I like to close on a positive note. There's a lot of unmet need in psychiatry. There are a lot of people suffering. major depressive disorder is the number one cause of disability adjusted life years lost in the industrialized world, and I think is going to be the number one cause in the world by 2030, according to the World Health Organization. So a lot of people suffering, a lot of them not getting the care they need, but we can help them, and we've made a lot of progress in the last 26 years, and here's to hoping for more. If I make it another 26 in the field.
We talk about this all the time, that there are really important unmet needs out there, people suffering, the need for us to do a good job. And if we do a good job, we're probably guaranteed work for the rest of our lives because there's still a lot left to do. So
Absolutely.
thank you, Dr. Detke, for joining us today and for this great conversation. It's been really interesting, and I hope to have another one with you again soon.
Great. Thank you, Dr. Zammit. Been a pleasure.